Now we are going to see next carbohydrate, disaccharides. Now disaccharides on hydrolysis gives 2 units of same or different monosaccharides. Disaccharides gives rise to 2 units of same or different monosaccharides on hydrolysis with dilute acids or specific enzymes. The 2 monosaccharides units are linked together by a ether oxide linkage, that is O linkage. We know that in ether there is presence of O linkage. Same O linkage or ether oxide linkage present in the disaccharides, which is termed as glycosidic linkage. This linkage is known as glycosidic linkage in carbohydrate chemistry. Glycosidic linkage is formed by removal of water molecule by reaction of 2 hydroxyl groups from 2 monosaccharide units. That is, when 2 monosaccharide units undergo reaction, then here removal of H2O takes place from 2 hydroxy groups of 2 monosaccharide to form a glycosidic linkage. At least one of the 2 monosaccharide unit must use its anomeric hydroxyl group in the formation of glycosidic linkage. That is, when glycosidic linkage formation takes place, at least one monosaccharide uses its anomeric hydroxyl group. 3 most common disaccharides are sucrose, maltose and lactose, and we are going to study the structure of these sucrose, maltose and lactose. First, sucrose. Molecular formula of sucrose is C12H22O11. Sucrose is dextrorotatory. This dextrorotation is plus 66.50. On hydrolysis with dilute acid or an enzyme called invertase, sucrose gives equimolecular mixture of D positive glucose and D negative fructose. That is, when sucrose, that is C12H22O11, when it undergo hydrolysis in presence of dilute acid or in presence of invertase, it is an enzyme, then here formation of C6H12O6, that is D positive glucose, and C6H12O6, that is D negative fructose. Since the levo rotation of fructose is minus 92.40, which is larger than the dextro rotation of glucose, that is plus 52.70, the hydrolysis product has net levo rotation. Now look here, fructose is levorotatory and glucose is dextrorotatory, but levo rotation of fructose is higher, that is minus 92.40, and that of that glucose, which is plus 52.70, and therefore the hydrolysis product has net levo rotation. Hence, hydrolysis of sucrose is also called inversion of sucrose, and the product is called invert sugar. Therefore, this sucrose is called as invert sugar. Actually, sucrose is dextrorotatory, but when it undergo hydrolysis, it forms fructose and glucose. Since the levo rotation of fructose is higher than that of dextro rotation of glucose, the resulting product has net levo rotation, and therefore this sucrose, which is dextrorotatory, when it undergo hydrolysis, it show levo rotation, and therefore it is called as invert sugar. Structure of sucrose contains glycosidic linkage between carbon number 1 of alpha glucose and carbon number 2 of beta fructose. Since the potential aldehyde and ketonic group of both the monosaccharides units are involved in the formation of glycosidic bond, sucrose is non-reducing sugar. Now look here, the structure of sucrose. Now this is the structure of sucrose according to Haworth projection formula. Now look here, this is alpha D glucose unit and this is beta D fructose unit. That is when sucrose undergo hydrolysis, it gives alpha D glucose and beta D fructose. Now here this alpha D glucose unit uses the anomeric hydroxy group, that is hydroxy group present at carbon number 1, and this beta D fructose uses its anomeric hydroxy group present at carbon number 2, and removal of H2O molecule takes place to form this O linkage, that is glycosidic linkage. It is called as glycosidic linkage. That is the linkage formed between the carbon number first or anomeric carbon of alpha D glucose and carbon number 2 or anomeric carbon of beta D fructose. It is called as glycosidic linkage. And this linkage, which is present in between it is formed in between alpha and beta hydroxy group of glucose and fructose respectively, and therefore it is called as alpha beta 1,2 glycosidic linkage. Alpha means OH group of alpha D glucose and beta means OH group of beta D fructose. 1,2 stands for linkage between carbon number 1 of glucose and carbon number 2 of fructose. Therefore, it is alpha beta 1,2 glucosidic linkage. Now, same structure is shown here. Now, here the linkage between this carbon number first or anomeric carbon of alpha D glucose and anomeric carbon of beta D fructose, that is carbon number 2, there is formation of COC linkage. This is called as alpha beta 1,2 glycosidic linkage. In this way, sucrose molecule is formed, or this is the structure of sucrose molecule. And when it undergo hydrolysis in presence of dilute acid or in presence of enzyme, then here formation of alpha D glucose and beta D fructose takes place. Now next example is of maltose. Maltose is a disaccharide having molecular formula C12H22O11. Maltose is a disaccharide made up of 2 units of D-glucose. The glycosidic bond in maltose is formed between carbon number 1 of one glucose ring and carbon number 4 of the other. The glucose ring, which uses its hydroxyl group at C1, is alpha glucopyranose. Hence, the linkage is called alpha 1,4 glycosidic linkage. The hemiacetal group at carbon number 1 of the second ring is not involved in glycosidic linkage. Hence, maltose is a reducing sugar. Maltose gives glucose on hydrolysis with dilute acid or the enzyme maltase. Now we are going to see the structure of this maltose according to Haworth projection formula. This is the structure of maltose. Now in maltose, this is alpha D glucose and this is also alpha D glucose. But here this glycosidic linkage is formed like this. That is alpha D glucose containing anomeric hydroxy group, and this is second alpha D glucose, but hydroxy group is used from the 4 number carbon atom, which is not anomeric carbon atom, and therefore this anomeric carbon atom of this maltose is free, and therefore it is called as reducing sugar. This is alpha 1,4 glycosidic bond or linkage in the maltose. Now lactose. Third example, lactose. Molecular formula of lactose is C12H22O11. It is a disaccharide present in the milk. It is formed from 2 monosaccharides unit, namely D-galactose and D-glucose. The glycosidic linkage is formed between carbon number 1 of beta-D-galactose and carbon number 4 of glucose. Therefore, the linkage in lactose is called beta-1,4 glycosidic linkage. The hemiacetal group at C1 of the glucose unit is not involved in the glycosidic linkage, but it is free and hence lactose is a reducing sugar. Now this is the structure of lactose according to Haworth formula. Now here it is beta-D galactose and this is beta-D glucose. Now we know that 1 number carbon atom is a anomeric carbon atom. Now in case of beta-D galactose, why it is called beta? Because hydroxy group is present on the beta side. Correct? Now here OH group of this beta-D galactose, that is OH group which is present at anomeric carbon or carbon number 1, is used in the formation of glycosidic linkage. Therefore it is beta. Therefore it is beta-D galactose, beta-1,4 glycosidic linkage. Now here why 4? Because the beta-D glucose uses hydroxy group present at the 4 number carbon atom, and here removal of H2 molecule takes place to form COC linkage, that is etheric linkage, oxide linkage, ether oxide linkage, that is COC linkage. Therefore it is called as beta-1,4 glycosidic linkage. Now in this case, the hydroxyl group present on beta-D glucose or hydroxyl group present at 1 number carbon atom or anomeric carbon atom of beta-D glucose is free, and therefore this lactose is called as reducing sugar. Now we are going to see polysaccharides. Now polysaccharides are formed by linking large number of monosaccharide units by a glycosidic linkage. Starch, cellulose and glycogen are the most common natural polysaccharides. Starch is a storage carbohydrate of plants and important nutrient for human and the other animals. Cellulose is the main constituent of cell wall of plants and bacterial cells. It is also main constituent of wood and cotton. Glycogen constitute storage carbohydrate of animals and is present in liver, muscle and brain. It is also found in yeast and fungi. Now first we are going to see starch. Starch is a polymer of alpha-D glucose. Starch has two components, namely amylose, that is 15 to 20% amylose, and amylopectin, that is 80 to 85%. Amylose is soluble in water and forms blue colored complex with iodine. That is, when amylose react with iodine, it forms blue colored complex. And it is soluble in water. It contains 200 to 1000 alpha glucose unit linked by alpha-1,4 glycosidic linkage, giving rise to unbranched chain of variable length. Now look here. Now it is starch is a polysaccharide and it is formed by using 1,4 glycosidic linkage of glucose. Now look here. This is first glucose molecule, this is second glucose molecule, this is third, this is fourth. Now when first molecule uses anomeric hydroxyl group in the formation of glycosidic linkage, at the same time hydroxyl group present at 4 number carbon atom is used for the formation of glycosidic linkage. That is, it is 1 alpha-1,4 glycosidic linkage. Same thing here also. Now here this, in case of second molecule of this chain, this glucose uses this hydroxyl group, that is anomeric hydroxyl group, with the third molecule which uses fourth hydroxyl group for the formation of this 1,4 glycosidic linkage. This is alpha 1,4 glycosidic linkage. That is, here this anomeric hydroxyl group which is present on alpha side is used in the formation of glycosidic linkage. It is called as alpha 1,4 glycosidic linkage. In this way, this amylose is formed. Amylose is a straight chain of alpha glucose. Now here in amylose, there are 200 to 1000 alpha glucose units are present. Now we are going to see amylopectin. Amylopectin is water insoluble component of starch which forms blue-violet colored complex with iodine. It is water insoluble, and when it reacts with iodine, it forms blue-violet colored complex. It is a branch chain polysaccharide. In amylopectin, chains are formed by alpha 1,4 glycosidic linkage between alpha glucose unit, whereas branches are formed by alpha 1,6 glycosidic linkage. Now look here, this is the structure of amylopectin. Now here this is alpha glucose, this is alpha glucose, this is also alpha glucose. All these are alpha glucose, but structure is branched. Now here if this is amylose chain, now here this amylose chain branch with the second amylose chain by 1,6 glycosidic linkage, that is alpha 1,6 glycosidic linkage. That is, here hydroxyl group which is present on 6 number carbon atom forms glycosidic linkage with the hydroxyl group of anomeric carbon atom of this amylose, and formation of alpha 1,6 glycosidic linkage takes place. That is, here amylose, number of amylose when undergo alpha 1,6 glycosidic linkage formation, then formation of amylopectin takes place. This is the structure of amylopectin. Amylopectin possesses alpha 1,6 glycosidic linkage as well as alpha 1,4 glycosidic linkage. This is the structure of starch. That is, starch possesses amylose and amylopectin. Now cellulose. Second example of polysaccharide is cellulose. Cellulose is a straight chain polysaccharide of beta glucose unit linked by beta 1,4 glycosidic bonds. That is, cellulose is a polysaccharide of beta glucose formed by beta 1,4 glycosidic linkage. Chemical hydrolysis of cellulose requires use of concentrated strong acid at high temperature and pressure. This implies that the beta 1,4 glycosidic bond is very strong and difficult to hydrolyze. Humans do not have enzymes which can hydrolyze this linkage. Hence, cellulose cannot be digested by human beings. It serves as a fibrous content of food, useful for bowel movement. Now look here, this is beta glucose, this is also beta glucose, this is also beta glucose. Why it is called beta glucose? Because the hydroxy group present at carbon number 1, that is anomeric carbon, is present on the left side in Fischer projection formula, and it is present on the upper side in Haworth projection formula. Now this hydroxyl group which is present on carbon number 1 or anomeric carbon forms glycosidic linkage with the second beta glucose molecule with the hydroxyl group of second beta glucose molecule present on the carbon number 4. Therefore, it is called beta 1,4 glycosidic linkage. That is, in cellulose, 1 beta glucose forms beta 1,4 glycosidic linkage with the second beta glucose molecule and so on, and formation of cellulose takes place. That is, when this cellulose undergo hydrolysis, it forms beta glucose, number of beta glucose molecules. Now last is glycogen. Glycogen is a animal polysaccharide which is present in animal. Glycogen has its structure similar to that of amylopectin, but it is more highly branched. Thank you.